energy source. It's a large molecule that we'll be talking about. ATP, the boxes are off a little bit here, but ATP is adenosine triphosphate. Triphosphate means 3 phosphates. It will react with water, and you will have a pyrophosphate pop off, which is just inorganic phosphate because it doesn't have a carbon to hydrogen bond. So we have ADP that's formed. So ADP has 1 less phosphate because it's diphosphate, and ATP is triphosphate. So that's how we release some energy. So we use this released energy to help us build polymers or to break polymers down into monomers, which is where we're starting to talk about these large molecules. So macromolecules are polymers. Macro means large, poly means many, mer means pieces, many pieces. That's going to be a very large molecule. And they're built from monomers. Mono means 1, mer means piece, 1 piece. Okay, so a polymer, many pieces, okay, is a large molecule consisting of many similar building blocks. And these small building blocks are called monomers, 1 piece. Okay, some molecules that serve as monomers have functions on their own, but they can also function to create a polymer, which will have its own capabilities. The synthesis and breakdown of polymers. Cells make and break down polymers by the same process. Okay, so we have a dehydration reaction, dehydration, to remove what? To remove water, dehydrate, okay? A dehydration reaction occurs when 2 monomers, 2 single pieces, bond together through the loss of water. So dehydration reaction is also called dehydration synthesis. Synthesis means to make or to combine. So through the removal of water, you're putting together monomers, building a polymer, okay? Polymers are disassembled to monomers by a hydrolysis. Hydro means water, lysis means to break. You are breaking a polymer by the addition of water. This is the opposite reaction, okay? A reaction that is essentially the reverse of our dehydration, which would be to remove water. If we're adding it, that's obviously the opposite. So these processes are facilitated by enzymes. Enzymes are a special class of macromolecule that we'll talk about. It's a protein, and their job is to help to speed up chemical reactions. So here's an example. For A, we have dehydration reaction or dehydration synthesis, which is synthesizing a polymer. So you can see that we have a short polymer and a monomer. And if you remove H2O, it's dehydration synthesis. You're removing water. You get a longer polymer with a byproduct of water. In reaction B, you have hydrolysis, hydro, water, lysis, to break. You are breaking down a polymer with the addition of water. So you can see that you have a polymer, and then you insert water, and you are splitting it into a short polymer and a monomer. Okay, the diversity of polymers. So each cell has thousands of different types of macromolecules, these large molecules we're going to talk about. Okay, so macromolecules vary among cells of an organism. They can vary within a species and even more between species. So all the cells of your body are composed of these 4 main macromolecules, but the concentrations are going to vary based on the different types of cells. And when you compare human cells to plant cells or bacteria cells, they're all made of the same basic 4 parts, mostly, but they're going to be in different concentrations based on the different environments and the different functions of those cells. Remember that the structure dictates the function. So if it's made up of different macromolecules, then it's got to have a different function. Okay, an immense variety of polymers can be built from a small set of monomers. Think of DNA as the example of that. We have trillions of blueprints of DNA that make different organisms, but it's made of the same 4 parts. So again, here we have 4 parts, our carbs, lipids, proteins, and nucleic acids that are going to make up all these different things that we need to function as living organisms. So there's going to be a lot of different polymers that can be formed out of those macromolecules. Carbohydrates serve as fuel and building material. So carbs are sugars. Carbohydrates include sugars and the polymers of those sugars. So it can be very simple sugars like glucose or fructose, which make sucrose, which is table sugar, all the way into starch, which are these huge long chains of glucose together, okay? The simplest carbohydrates are called monosaccharides. The simplest carbohydrates are called monosaccharides. Mono means what? It means 1. Saccharide means sugar. So the word literally means 1 sugar. That's obviously going to be the simplest or the smallest sugar. It is just one ring that we're talking about. We'll look at the structure in a second. Carbohydrate molecules, macromolecules can be called polysaccharides. Poly means many, and now we know saccharide means sugar, many sugars. Okay, polymers composed of many sugar building blocks. So a monosaccharide is one ring structure for the simple sugar, and we have our polysaccharides are going to be polymers of these monomers, right? Okay, so monosaccharides have the molecular formula that is basically CH2O. So basically that's saying that there is twice as much hydrogen as carbon and oxygen. So carbon and oxygen have to be equal because CH2O means that we have a one carbon, two hydrogens, one oxygen. It's called a 1 to 2 to 1 ratio because there's one carbon for two hydrogens and one oxygen. Okay, so the best example of that is glucose. Glucose is written as C6H12O6. Again, carbon and oxygen are equal, and there's twice as much hydrogen. It's a 1 to 2 to 1 ratio. This is a golden rule for carbohydrates. If it's going to be considered a carb, it has to have a 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. Close up, moving on. Okay, in the straight structures of cellulose, the hydrogen atoms on one strand can form hydrogen bonds with the OH groups on the other strand. Parallel cellulose molecules held together this way, groups called microfibrils. Okay, this creates the strong building material for plants. It's got a very rigid structure. It's very tough. Okay. Enzymes that digest starch by hydrolyzing these alpha linkages can't hydrolyze the beta linkages in cellulose, which is important because you probably don't want to break down, the outside of your own cell, okay? Cellulose in human food passes through the digestive tract. Why do you think when you eat corn, you get the corn poops? You know what I mean. You know exactly what I mean, okay? We can't digest cellulose. Think about it, all right? Okay, you with me? You caught up? Okay, cool. So some microbes use enzymes to digest cellulose. We don't, okay? It's thought that our appendix might have been an area in our ancestral history of evolution, that the appendix, which, you can get taken out now and you don't die or anything, you're totally fine because it's a structure we no longer need called the vestigial structure. It was once thought that this appendix had a function, and it was to break down cellulose because chimpanzees and other apes and things like that obviously eat a lot of foliage and would need to break that down for nutrients. But our diet as humans has changed quite a bit since then. So it's hypothesized that that's where we were able to break down these beta linkages once upon a time. Many herbivores, from cows to termites, have symbiotic relationships with these microbes. So basically, these microbes that can break down cellulose live in the intestines of these organisms that are herbivores that would have to break that down. This word looks like it's pronounced chitin, but it's pronounced chitin, like go fly a kite. Okay, chitin. Another structural polysaccharide is found in the exoskeletons of arthropods. It's also found in fungi. So chitin also provides the structural support for the cell walls of many fungi. So like mushrooms and things like that have a cell wall made of chitin. Not chitin, chitin. Okay, our next class of macromolecules, all of Okay, our next class of macromolecules, all of that jazz was about carbohydrates. So our next topic is lipids. Lipids are fats. So we talked about sugars, now we're going to talk about some fats. So lipids are a diverse group of hydrophobic molecules. Now we just learned that word last lecture, hydro, water, phobic, fearing. This means that they're not going to dissolve in water. They don't like water. They don't want to touch water, okay? So lipids do not form true polymers. They just kind of get close together. They don't actually form linkages like those glycosidic linkages between the monomers to make a polymer. They just kind of huddle up. Okay, and it's because of the chemistry of a lipid itself, which we'll look at in a minute, okay? They don't actually bond together to form true polymers, but some of them are quite large themselves, and then they aggregate. They kind of get close to each other to form something that looks like a very large lipid, but it's a whole bunch of little lipids. Okay, so the unifying feature of lipids is having little or no affinity for water, hence hydrophobic, okay? So lipids are hydrophobic because they consist mostly of hydrocarbons. So carbon to hydrogen bonds, which form nonpolar covalent bonds. Now, why is it important that we specify nonpolar? What's water? Water is polar because it has an oxygen with our two little hydrogens. The oxygen is slightly negative. The hydrogens are slightly positive, which means it has a pole on either end of the molecule. It is polar. Okay, a hydrocarbon is not positive or negative. It's neutral. Okay, there's no slight charges anywhere in there. So they are nonpolar. And what was the rule when we talked about solutions and how water is a universal solvent? What did we say? Like dissolves like. If water is polar, it will dissolve other things that are polar. Okay? With lipids, they are nonpolar. So if you add lipids to water, will it dissolve? No, absolutely not. Okay? They have little or no affinity for water. Most biologically important lipids are fats, like phospholipids, and our steroids, which we're going to talk about, like hormones, cholesterols, things like that. Okay? We have fats for like insulation purposes and energy storage. Our phospholipids are very important for every living organism because that is the main component of the cell membrane. And then our steroids, like I said, hormones and our cholesterols are important for maintaining balance in the body as well as maintaining the the lack of rigidity of our cell membranes. Okay, so lipids, fats. So fats are constructed from two types of smaller molecules called glycerol and fatty acids. Okay, so we're going to have a glycerol backbone. Typically, we're going to be talking about triglycerides that have 3 fatty acid tails. Okay, glycerol is a 3-carbon alcohol. OL tells you it's an alcohol with a hydroxyl group attached to each carbon. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. Here's what that looks like. Here's your glycerol. Skeleton. carboxyl group attached to a long carbon skeleton. Here's what that looks like. Here's your glycerol backbone. And then here we have a fatty acid. This one is palmitic acid. Backbone. And then here we have a fatty acid. This one is palmitic acid. Okay, so you see that you have your carboxyl group here, you have your OH group here. This is a dehydration reaction because you're removing water in order to snap these two together. Okay, but if you look here, this little CH little tail, this fatty acid tail here, this is going to be like no charge. This is nonpolar. This is what makes the molecule nonpolar, why it doesn't dissolve in water. Okay, now if you do 3 of these dehydration reactions, you create something called a triglyceride. Tri means 3, 1, 2, 3, and the glycerol backbone. It is a triglyceride. Fats separate from water because water molecules hydrogen bond to each other to exclude the fat because they don't want to hydrogen bond to these little fatty acid tails. Okay, they want to hydrogen bond to each other because they are polar. In a fat, the 3 fatty acids are joined to glycerol by an ester linkage, which it shows you right here, ester linkage, creating a triacylglycerol or a triglyceride. We will always call it a triglyceride for the intents and purposes of my class.
Anh Nôm, anh Nôm.ไอ้นมเอ็มไม่คุยกับพี่ว่ะน้อง
残り1名。A でキャリアダウン。
เจอตรงนี้
ナイス。
So fatty acids vary in length. Like we looked at this one was called palmitic acid. There's other ones. So that's just the number of carbons. Okay, so they're named for the number of carbons in their hydrocarbon fatty acid tails. And then the number and locations of the double bonds, which leads us to saturated and unsaturated.
B でキャリアダウン。残り1名。
まだやられちゃいないよ。 組むのよ。でも私は闇など恐れないわ。ぶっ飛ばしてやる。
最後の一人だ。A でキャリアダウン。
私たちは家族。負けるわけないって。装備くれない？
In hydrogen, which means that's missing. That's here and a little hydrogen floating here. So
A でキャリアダウン。
銃がある。行け。スパイク設置。行け。グレネード。ここ。
視界を
敵残り1名。
アルティメット準備完了。
おやつにしよっか。アルティメット準備完了。
it's not saturated in hydrogen, which means it's missing hydrogen. But can you just not have a bond? Not in this case. Okay, what's happening is that you have a double bond, one or more, which means that really you're eliminating 2 hydrogens for each double bond. Okay, we're going to look at some pictures of that. But when it says saturated fatty acid or an unsaturated fatty acid, and that means nothing to you, what is it saturated in? Think about what it actually means. Okay, it means that it's saturated in hydrogen, which means you can't have a double bond. If it is unsaturated in hydrogen, it means that it is missing, quote, missing some of its hydrogen. So let's look at that. So saturated fat is going to have all, look at these little tails, okay? That's exactly the same as this. It's our carbon to carbon to carbon to carbon with our little hydrogen tails here, right? Because we have 4 bonds that each one of these carbons can make. 1, 2, 3, 4. You actually don't write all this out. You would never do that, okay? It's this little zigzag. So this means that you have a carbon to a carbon to a carbon, but remember that there's a little hydrogen floating here and a little hydrogen floating here. So really you have 1 bond here, 1 bond here, 1 to hydrogen, 1 to hydrogen. This carbon has 4 bonds, even though you just don't write it. Again, you should have learned this in Gen Chem. Okay. So this is a saturated molecule because it is saturated in hydrogen. Okay, these are things that are more solid at room temperature. Unsaturated. Unsaturated means that we're missing, quote, missing what? We're missing hydrogen. It's right here. Okay, we have a carbon to carbon double bond, which means that this carbon is connected to a carbon here. So that's 1 bond. 2, 3. It is double bonded over here. So we only have 1 more little hydrogen. Well, how many hydrogen should be there? 2. right? But now we used one of those hydrogen bonds basically to put it here to have a double bond. And notice how it changes the shape. This shape is straight, this shape is straight, this shape is now bent. What if I had 4 double bonds? You'd have a whole kind of like big curve, right? Now, if you have a whole bunch of these molecules stacked on top of each other, would it be able to pack as tightly as these molecules stacked, unstacked, unstacked, unstacked on top of each other? No, it would not be able to get as close, because if you put another one of these underneath here, it can't get very close. It's going to run into it, so it's got to leave some space. That's why unsaturated fats are liquid at room temperature. But our saturated fats, because there's no curve, it's all straight, they can just like bricks. You can just stack a bunch of these on top of each other. That's why they are solid at room temperature. It's important that you understand the differences there. Okay, so fats are made from saturated fatty acids called saturated fats, and they are solid at room temperature, which we just talked about. These are mostly animal fats that are saturated. Because think about like butter, lard, these are things that come from animals, whereas like olive oil, olive is, I guess it's considered a fruit. It comes from a tree, okay? So that is a plant. So most of our saturated fats come from animals, and then fats made from unsaturated fatty acids are called unsaturated fats or unsaturated oils, and they are liquid at room temperature. Remember why? It's because of those, the shape here of our double bonded. It's because of those, the shape here of our double bonded carbons. Our double bonded carbons change the shape. You can't pack them tightly together. So plant fats and fish fats are typically unsaturated. The major function of fats is to store energy. I think about carbs. Carbs are what? Carbs are sugar. Sugar, short-term energy. Okay, lipids are fats. Lipid, long-term. You notice the alliteration? Sugar, short-term, lipid, long-term energy storage, because you're storing a large amount of energy, because these like longer structures, right? These have a lot of different bonds in them. It's a very long structure. They're going to have a lot of energy that they're storing for us, okay? Fat is a compact way for animals to carry their energy stores with them. Okay, this is especially important when you have animals that are trying to load up for hibernation, or animals that live in cold environments, right? They're going to need to have that fat to store the energy with them and to act as insulation in some cases. Phospholipids. It's a lipid, which means it's a type of fat. Phospho means it has a phosphate slapped on it. So it is a lipid with a phosphate group attached. This is one of the most biologically important structures. In a phospholipid, 2 fatty acid tails and a phosphate are grouped together with the attachment of a glycerol. Okay, so instead of a glycerol with 3 fatty acid tails to make a triglyceride, we're making a phospholipid that has a phosphate, has a glycerol, and 2 fatty acid tails. So the 2 fatty acid tails are, sorry about that, they are hydrophobic. Again, they're hydrocarbons. We know that all hydrocarbons are nonpolar and therefore are hydrophobic. Okay, but a phosphate group that's attached to it forms a hydrophilic head, because a phosphate group is a polar group, okay? And that is hydrophilic, hydro, water, philic, loving. It's a water-loving phosphate head because it's polar, and so is water, okay? So phospholipids are the major
安らかに。
B でキャリアダウン。あと30秒。
Major constituent of the cell membrane. Okay, this is most of what the cell membrane is made out of. And that's obviously very important to everything that is alive, because every single cell that has been or will be alive contains phospholipids. Okay, so here's the structure. So you can see that we have the hydrophilic head and the hydrophobic tails. You can see that there are two fatty acid tails. You can see that we have a phosphate group. We have a glycerol backbone. Here's what it looks like to me. I like to think about it like it's a little lollipop with two little sticks, because when I draw it, because I'm not an artist, that's what it looks like. I draw a circle, I draw two little legs, and usually one of the legs is a little wonky. It's like it's doing the stanky leg, right here. You know, straight leg, and then like, whoa, what's that mean right here? This little whoop. Why does it look like that? Why wouldn't it be straight? What happened right here? If you said carbon-carbon double bond, you win a prize. You're correct. Okay. So this is what a phospholipid bilayer would look like. What's a bilayer? Two layers. Two layers of what? Phospholipids. What's a phospholipid? This little guy. You got two layers of them. Why are all the tails touching? Why isn't it like this orientation with another row of the same orientation? Like this little like blue-gray, orangey, blue-gray, orangey. Why is it blue-gray, tail, tail, head? Why does it do that? Head, tail, tail, head. What is this word right here? It's crossed out because of the little arrow, but hydrophobic, water fearing. What do you think this blue stuff is? If you said water, you're right. Okay. So the hydrophilic heads will touch the water. The hydrophobic tails do not touch the water. They're on the inside of this bilayer. This is the structure, the basic structure. There's a lot more to it, but this is the basic structure of every cell in your body, in every other body, and every bacteria and plant. Okay? This is the basic structure of the phospholipid bilayer, that is also called the cellular membrane. So when phospholipids are added to water, they self-assemble into this bilayer. They just do it on their own. No one's had to say, okay, line up, come here, we gotta have a head this way, tail this way. No one tells it what to do. It just does it. How does it know to do it? Because of the properties, the chemical properties that we learned about, the hydrophobic tails and the hydrophilic head. If it knows that the tails can't touch water, you gotta have two layers. You're not gonna make 6 layers. That wouldn't make any sense. You have to have 2 layers to protect those tails. You can't have 3 layers because where are the other tails gonna go, right? And actually, spontaneously, phospholipids will make something called a micelle, which is a circular sphere. That's how we get our membranes. Okay, so when phospholipids are added to water, they self-assemble into a bilayer with the hydrophobic tails in the middle because they can't touch the water. Okay, this feature of our phospholipids results in a bilayer arrangement for our cell membranes, like we just talked about. So steroids are actually still part of lipids. We've not moved on yet. We are still on number 2, lipids. Okay, we are, something I forgot to point out that I'm going to do real fast. All of our lipids, right? This is a lipid. This is a phospholipid. So this is a phosphate group right here. We're going to pretend like he's gone for a second. Okay, just bear with me. We have C, H, and O. CHO. What do we have for sugars? CHO. What was special about C, H, and O and sugars? Had to be a 1 to 2 to 1 ratio. If we have CHO, C, H, and O, and it's not a 1 to 2 to 1 ratio, it's a lipid. Okay, this is a fancy lipid called a phospholipid, so it has a phosphate. Okay, but like, it's C, H, and O. I forgot to mention that, I'm sorry. Okay, It's CH and O. I forgot to mention that. I'm sorry. Okay. We're here. So steroids are also lipids because they contain CH and O. CHO. Is it a 1 to 2 to 1 ratio? No, because then it would be called a carbohydrate. Okay? But steroids are just a class of
A でキャリアダウン。最後の一人だ。
lipids.
スパイクを
So
設置。
steroids are lipids that are characterized by a carbon skeleton. Skeleton consisting of 4 fused rings. And we know that a ring is actually more of a hexagon because we just looked at those when we talked about sugars. Okay? Cholesterol
Reload.
スパイクを設置。残り1名。
さあ。
最後の一人だ。
狙いを定めて突入する。わかった？
凪の中で私は見る。嵐の中で私は戦う。グルネード。壁を展開。排除した。
B でキャリアダウン。
するわ。
残り1名。
グルネード。
ここ。ここ。スパイクを設置。
ここ。
is an important steroid, and it's a component in the cell membrane of all animals. Okay, well, other membranes too, but all cell walls pretty much. We have cholesterol that is a very important component of our cell membrane that now we kind of know looks like this phospholipid bilayer. Okay, so although cholesterol is essential in animals, high levels in the blood may contribute to cardiovascular disease because it'll cause it to clump up in your arteries and veins. Okay, but the 4 fused rings, they're fused because they're all touching, okay, is the basic structure of a steroid. Okay, and so when you see these little cholesterols, because cholesterol is a type of steroid and a steroid is a type of lipid, so technically a cholesterol molecule is also a lipid. Okay, this is just a special kind of lipid, looks very different from other lipids. Okay, but you will see these little like goldeny rings in the middle of a membrane, and that's what we're talking about. We don't draw the rest of this crap because it takes too long, okay? We just draw these little rings in the middle of the phospholipid bilayer to show you that there is still cholesterol present. Okay, proteins. Number 3, proteins. Before I forget, we're dealing with C-H-O-N, CHON, C-H-O-N, CHON, and sometimes S. C-H-O-N, sometimes S, okay? So those are the elements that we're dealing with. We went CHO with a 1:2 to 1 ratio, then we went CHO, lipids, with no special ratio, and now we're on CHON, CHON, protein, CHON. They both end with the letter N, and stands for nitrogen in the case of an element, okay? So we have carbon, hydrogen, oxygen, nitrogen, sometimes sulfur.
こスパイクを設置。じゃあね。敵残り1名。アルティメットいけるよ。最後の1人だ。クラッチ。ペンキよし、爆薬よし、ブレーキなし、アクセル全開。狩りの時間よ。
敵残り1名。
終了。
Okay, so protein, Chan.
誰か買ってもらえますか？
人暴れしよっか。
うせな。
爆弾起動。パーティータイム。グレネード。
ここよ。
いけ。
次はドイツだ。
リロード。
スパイクを設置。
宗教不足。まだ終わりじゃないわ。
方面撃破。敵残り1名。
Sometimes S. CHON, sometimes S. Okay? So the
爆弾から逃げたところを狙い撃ち。楽勝でしょ。
誰か買ってくれない？
爆弾行くよ。グレネード。いけ。
those are the elements that we're dealing with. We went CHO with a 1 to 2 to 1 ratio. Then we went CHO lipids with no special ratio. And now we're on CHON. CHON, protein. CHON. They both end with the letter N. N stands for nitrogen in the case of an element. Okay, so we have carbon, hydrogen, oxygen, nitrogen, sometimes sulfur. Okay, so protein, CHON. So proteins. Proteins include a diversity of structures. They make up pretty much all the structural components of your body, resulting in a wide range of function. Because why? Because what is the favorite theme here? Structure dictates function. That's correct. Structure dictates function. So we have a large diversity of structures, which means we have a large range of functions. Okay, so proteins account for more than 50% of the dry mass of most cells. We know that it's mostly water, so if we take all that water out, 50% of what's left over is going to be proteins. That's a lot of protein, okay? Most proteins, well, their structure is going to vary widely, which means that their functions are going to vary widely. But they can function in a couple of these things here, in defense, like for your immune system, storage, transport, cellular communication, which we'll talk about in chapter 5, movement, which we'll talk about in chapter 4, and structural support. There's a whole chart here in your textbook that looks something like this. It says, like, these are all the things that our proteins can do. They can have enzymatic proteins. We can have defensive proteins, storage proteins, transport proteins. There's a bunch, obviously, and this is just a little snapshot. We will have a whole chapter over enzymes, okay? This is largely what we're talking about when we talk about proteins, that they are enzymes. But they do all these other really cool things too, and those are all equally as important. Okay, so life would not be possible without enzymes. Like I said, it's listed first. We're going to talk about it mostly because that's really what we're interested in for the intents and purposes of a protein right now. So life would not be possible without enzymes. Enzymes are another word for proteins, but enzymes specifically act as
マッチ
catalysts.
ポイント
A
負
catalyst
けないって任せときな。消えぬ。
A でキャリアダウン
見えないでしょ。いけ。見えないでしょ。
残り1名 A でキャリアダウン
限界など耐えるのだ。
誰か買ってもらえますか？誰か買ってもらえますか？ありがとう。今回やめとく。
次はドイツだ。
スパイクを設置 残り1名
回復まだ無理。
パーフェクト
次はどう来るの？特定のパターン
speeds up a chemical reaction. So an enzyme is a catalyst, and a catalyst speeds up a chemical reaction. But how does it do it? An enzyme is a catalyst that speeds up chemical reactions by lowering the activation energy. You will need to know that definition, which is why I just said it slowly for you, okay? So if we know that they speed up chemical reactions by lowering the activation energy, this means that you don't have to build up or make a whole bunch of energy in order for a reaction to proceed. It actually requires a lot less energy. So polypeptides. Poly means what? Many. Peptide. Spoiler alert, it's the name of a bond. Okay, many peptides is another name. So polypeptides is another name for a protein. A protein and a polypeptide are the same thing, okay? So polypeptides are unbranched polymers built from the same set of 20 amino acids that we have in our bodies. Okay, a protein is a biologically functional molecule that consists of one or more polypeptides. Polypeptide, many peptides, which is the name of a bond, is a protein. Okay. Amino acids. Amino acids
B でキャリアダウン最後の1人だあと30秒残り1名 あと10秒
is a protein. Okay. Amino acids. Amino acids are the monomer of a protein. Okay, lipids are weird that there's no true polymer. Whatever. Okay. Our monomer for carbohydrates was what? A monosaccharide. Okay, one piece of a protein is called an amino acid. Okay, and my really dumb way to remember that is a protein. Pretend it's T-E-E-N, like a teenager. What do teens like to do in school because they want to be intelligent and go off to awesome colleges or other careers that they love? They want to make A's, the letter, not the disease, right? Teens like A's, amino acids. That might be dumb, and you might be laughing at me or think that's stupid, but come test time, if you remember that, you're welcome. Remember the dad jokes and the lame excuses here. Okay, so amino acids are the monomer of a protein. Okay, they are organic molecules, which we learned means that they are carbon-containing molecules. They were not raised on a farm. Okay, with carboxyl and amino groups, which again, you saw at the beginning of this and also in the last chapter a little bit at the beginning, are functional groups, carboxyl and amino groups. So amino acids differ in their R group, which I'm hoping there's a picture. Beautiful. Okay, so this is the basic structure of an amino acid. Is this particular structure an amino acid? No, because there is no element called R. R is a placeholder. It's like this right here. It could be 20 different things. There are 20 different amino acids that we use in our bodies. So there are 20 different things that could be right here. So we just call it R because it's a little placeholder. Okay? But we have an amino group and we have a carboxyl group and then something that's coming out over here, one of 20 options. That would make an amino acid. This is the basic structure of an amino acid. It's just missing its R group. It hasn't been specified. So this is the basic skeleton of an amino acid because we have an amino group, a carboxyl group, and an R group placeholder. Here are the 20 amino acids. If you go and take this at a major university like UT, Texas A&M, Rice, you will need to memorize all of these. You will need to memorize if they are nonpolar, if they are polar, if they're charged, if they're not charged, acidic, basic. You will need to be able to draw all of these from memory. For my class, you will not, but what I want you to start to understand is all of the naming, right? So like glycine, GLY. It's typically the first three letters. There are some exceptions. Alanine, ALA. This will be important later. But for the most part, it's the first three letters. That's what I need you to know. Okay. Leucine, isoleucine, methionine, phenylalanine, tryptophan, proline. They got some weird names. But if you look, the abbreviations are typically the first three letters. Typically, there are some exceptions. Okay. So polypeptides, again, just remember that that is another word for proteins. Okay, so amino acids are linked together by peptide bonds. Think about a pearl necklace, like beads on a string. You got all those little balls and you got a string running through it, right? Each one of those little beads, each one of those little pearls is actually an amino acid. And the string running through it is a whole bunch of different peptide bonds. Hence, polypeptide, many peptide bonds between amino acids makes a protein. Okay, a polypeptide is a polymer, so many pieces of amino acids. Amino acids are the subunits. Okay, polypeptides range in length from a few to more than 1,000 monomers of these amino acids. Each polypeptide has a unique linear sequence of amino acids with the carboxyl end, which is called the C terminus because the carboxyl group contains carbon. Okay, and the amino end, which is called the N terminus because it contains nitrogen. That's where the N comes in CHON, sometimes S. Chon, that's where the N comes in, the N terminus, which you saw here, the C terminus and the N terminus. Comes in, the N terminus, which you saw here, the C terminus and the N terminus. Okay, so this is what it looks like here. So you have the N terminus, you have the C terminus, you have the N terminus, you have the C terminus, you have the N terminus, you have the C terminus. Okay, Chon, that's where the N comes in, the N terminus, which you saw here, the C terminus and the N terminus. Okay, so this is what it looks like here. So you have the N terminus, you have the C terminus, you have the N terminus, you have the C terminus, you have the N terminus, you have the C terminus. Okay, here we have a dehydration reaction when we're removing water and we are adding a new peptide bond. So this is an amino acid. This is an amino acid. This is an amino acid. Notice that all the side chains are different. That's what that R group used to be, right? So these are actually amino acids here. Okay, so protein, structure, and function. The main theme of biology, structure and function. Okay, a functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. Proteins are crazy. They're finicky. They're complicated, but they do really important things, so they kind of deserve to be complicated. There's a whole bunch of protein folding rules that come into play. Here you can see we have like beta sheets and alpha helices, which is the secondary structure of a protein. The primary structure of a protein is beads on a string, kind of like just an amino acid sequence. This would be more of like the secondary structure. Okay, and when we get into tertiary is when you start having, is it going to show any pictures? Okay, we'll get there in a second. Tertiary and quaternary are going to be the advanced and fully functioning varieties of our protein structure, and we'll talk about that in just a second. The sequence of amino acids determined genetically, because you get your sequence of amino acids based on the sequence of your DNA, right? The nitrogenous bases in your DNA leads to a protein's three-dimensional structure, which is the actual working protein. A protein structure determines its function. The structure determines its function. We know that. This is not news, okay? 4 levels of protein structure. Proteins are diverse, and they share 3 superimposed levels of structure called primary, secondary, and tertiary. There's actually one that comes after that, the 4th level, quaternary, which arises when a protein consists of more than one polypeptide chain. I just kind of want you to be able to recognize primary and secondary are the important ones for my intents and purposes. Okay, understand that primary and secondary are also not functioning yet. You need to be a tertiary structure or have more than one tertiary structure together to make the quaternary structure. Okay, so the primary structure of a protein is its unique sequence of amino acids. Okay, the secondary structure is found in most proteins and consists of coils and folds called beta sheets and alpha helices. The tertiary structure is determined by the interactions. It's a further folding mechanism determined by the R groups. We looked at polar, non-polar, acidic, basic. That will determine the folding structure, and then the quaternary structure results from the interactions between several of these chains put together. So you can see here, this is the pearl necklace, the beads on the string. Okay, glycine, proline, threonine, glycine, threonine, glycine, glutamine, right? You have all of these amino acids put together, and in between all of them, you know that you have a peptide bond. Okay, that's primary. Secondary, we have alpha helices or alpha helix and a beta sheet, a beta pleated sheet. Then when those things interact based on their R groups, you're going to have the tertiary structure looks like this, where the secondary structure is still obvious. And then if you have more than one tertiary structure put together, you can have a quaternary or quaternary structure here to have a fully functioning protein. This can also be a fully functioning protein depending on the protein. Okay, there's van der Waals interactions, which was from the last lecture, one of the interactions between the R groups that kind of determine what these shapes actually look like. Okay, here you can see a bunch of alpha helices for collagen. Our heme in our, the heme group in our red blood cells contains a whole bunch of different subunits. So it's a whole bunch of polypeptide chains put together. And you can see, again, these helices are very prominent. Okay, sickle cell disease is a change in primary structure, which is talking about our amino acids, which is derived straight from the DNA. So it is it's a genetic thing. So
C に敵がいる。
最後の一人だ。スパイクを設置。パーフェクト。
油断しないで。戦場は常に変化する。ターゲットダウン。バリア設置。
狩りの時間よ。
アルティメットいけるよ。
敵残り1名。
Here you can see a bunch of proline 3 and between the R groups. Just kind of want you to be able to recognize primary and secondary or the
ここよ。
スパイクを設置。
50人だろうが関係ねえ。行くぞ。
気をつけて。バリア設置。さよ、ここが奴らの墓場だ。アルティメット引き寄せる。助けなど来ない。
敵残り1名。A でキャリアダウン。
リロード中。銃があるよ。次はどう来るの？特定のパターンが？
ここよ。
The important ones for my intents and purposes. Okay, understand that primary and secondary are also not functioning yet. You need to be a tertiary structure or have more than one tertiary structure together to make the coordinate. You need to be
誰かお金余ってない？
really important things, so they kind of deserve to be complicated. There's a whole bunch of protein folding rules that come into play. Here you can see we have like beta sheets and alpha helices, which is the secondary structure of a protein. The primary structure of a protein is beads on a string, kind of like just an amino acid sequence. This would be more of like the secondary structure. Okay, and when we get into tertiary is when you start having, we're going to show any pictures. Okay, we'll get there in a second. Tertiary and quaternary are going to be the advanced and fully functioning varieties of our protein structure, and we'll talk about that in just a second. The sequence of amino acids determined genetically, because you get your sequence of amino acids based on the sequence of your DNA, right? The nitrogenous bases in your DNA leads to a protein's three-dimensional structure, which is the actual working protein. A protein structure determines its function. The structure determines its function. We know that. This is not news, okay? 4 levels of protein structure. Proteins are diverse, and they share 3 superimposed levels of structure called
ここよ。ここよ。最後の一人だ。パーフェクト。命乞いさせてやるわ。
primary, secondary, and tertiary. It's actually one that comes after that. The fourth level, quaternary, which arises when a protein consists of more than one polypeptide chain. I just kind of want you to be able to recognize primary and secondary are the important ones for my intents and purposes. Okay, understand that primary and secondary. Secondary are also not functioning yet. You need to be a tertiary structure or have more than one tertiary structure together to make the quaternary structure.
壁を消すわ。スパイクを設置。敵残り1名。
いけるわ。バリアよ。
スパイク解除。
楽に行かせてやれよ。せめてもの情けってやつだ。
やるじゃない。
せいぜい死ぬなよ。
A でキャリアダウン。敵残り1名。
地獄行きだ。
攻守交代前、最終ラウンド。
最高の装備を買って手遅れになる前に。
アルティメットいけるぜ。
A でキャリアダウン。
逃がさない。敵を倒した。音を聞く。
A でキャリアダウン。
まだ終わりじゃないわ。
動きは
あと30秒。
Okay, so the primary structure of a protein is its unique sequence of amino acids. Okay, the secondary structure is found in most proteins and consists of coils and folds called beta sheets and alpha helices. The tertiary structure is determined by the interactions. It's a further folding mechanism determined by the R groups. We looked at polar, nonpolar, acidic, basic. That will determine the folding structure. And then the quaternary structure results from the interactions between several of these chains put together. So you can see here, this is the pearl necklace, the beads on a string, okay? Glycine, proline, threonine, glycine, threonine, glycine, glutamine, right? You have all of these amino acids put together, and in between all of them, you know that you have a peptide bond, okay? That's primary. Secondary, we have alpha helices or alpha helix and a beta sheet, a beta pleated sheet. Then when those things interact based on their R groups, you're gonna have the tertiary structure, looks like
一人だ。パーフェクト。
この仕事を選んだことを後悔させてやる。
リロード。
スパイクを落とした。引っ込んでろ。スパイクを確保。デコイだ。
Ay, Ayun, naka-on na. Putang na. Bawa naman ito.
逃がさない。
壁を展開。
Doon sila sa flank ko eh.
注意して。
敵残り1名。
Ah, ganun ba yan?
まだ使えない。
Mada tsukawanai yo. Sonda makoto man.
私っぽくないこと言うけど、多分力を合わせるんだよ。
誰かお金余ってない？
リロード中。
アメリカに行っちゃったね。いいよ
誰かお金余ってない？
いいよ、そんなことも。Vậy đâu mà đua xe. Oh
キャリアダウン。
no.
ネット展開。バリア設置。
最後の一
Sarap
人だ。
ng buhay pala. Mhm.
敵をかき回し生き残りがいたら片付けとく。
ここよ。ここよ。
始まる。
かあるもんね。おいおいおい。
逃げても無駄。
行くぜ。
最後の一人だ。
Like this, where the secondary structure is still obvious. And then if you have more than one tertiary structure put together, you can have a quaternary or quaternary structure here to have a fully functioning protein. This can also be a fully functioning protein depending on the protein. Okay, there's van der Waals interactions, which was from the last lecture, one of the interactions between the R groups that kind of determine what these shapes actually look like. Okay, here you can see a bunch of alpha helices for collagen. Our heme in our the heme group in our red blood cells contains a whole bunch of different subunits, so it's a whole bunch of polypeptide chains put together, and you can see again these helices are very prominent. Okay, sickle cell disease is a change in primary structure, which is talking about our amino acids, which is derived straight from the DNA, so it is it's a genetic thing. So primary structure is a sequence of amino acids on the polypeptide chain. A slight change in this primary structure can affect the protein structure and therefore its ability to function, because structure dictates function. If you change the shape, you have now changed the function. So sickle cell disease is an inherited blood disorder that results in from a single amino acid substitution. So this just means
ここよ。
もういいよ。
ここよ。
毒の壁よ。
フェイク
スリッティ。
銃を買うならちゃんと使えよ。戦い続けろ。
ここよ。
たとえ一人でも
誰か買ってもらえますか？
止まるわけにはいかねえんだ。
誰か買ってもらえますか？
余ってない？
遊びたいのか相手にな。
Grabe, nag-connectan na gamit akong up. Fuck. Yawa.
キルを
Sige, isa pa.
アルティメットいける。解除した。
Isa pa.
動きを封じる。
No! May subgun shot, Kasan! Walang ganyan ang aking agent. Left ni mo lagi.
残り1名。壁を消すわ。マッチポイント。あと一息ね。痛めつけてやる。
必要なものがある。
ダメよ。
You're welcome. You're welcome.
全てを奪ってやる。
Ano siya icon ba? Gusto ko na siya icon.
Oh, 終わりだよ。 何もない。
行くぜ。
敵残り1名。
アルティメットいけるぜ。
Defuse, 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 defuse.
アタッカーの勝利。
じゃあな。この世の終わりでまた会おう。
One of those amino acids, one bead on the string, has been substituted for a different one, and now the protein in hemoglobin, this structure, has changed. Different one, and now the protein in hemoglobin, this structure, has changed, which means its function will change. It has a lower affinity for oxygen. Okay, so it also, you can see here's the original shape, here's the sickle cell. It has a different shape. Okay, because of the different here, instead of glutamine, we inserted a valine. So just one. So you can see how much impact just one of these amino acids would have on the whole thing. Okay, you still made a red blood cell, but did it make a functioning red blood cell? Not so much, right? You can see all of the different structural differences based on the different shapes. Okay, so what determines protein structure? Okay, in addition to the primary structure, which we know is the amino
nice you. Ayo. Gusto ka magdula Robloxon niya? Dili ni mo.
Okay.
Really? Na naka-tablet?
Yeah. Yeah.
Okay, mag-chat lang ako ha.
Okay.
Okay, bye-bye.
Bye-bye.
Kiss ko ya.
No.
Ah. Saan nga si Tulis? Gusto na ko sila sa bayon. Igis pare.
Amino acid sequence, physical and chemical conditions can affect the structure.